Thank you. So it's my distinct pleasure today to get to introduce Holly Holmes to present her master's thesis work on Harlequin Ducks in the Northern Rockies. And before I tell you anything about Holly, I want to take a moment to extend a welcome to the huge audience online. There are many, many collaborators, volunteers, friends, and family joining that way who aren't here in this room, and this room is pretty full. Holly's work has been hugely collaborative, starting in Glacier National Park and with the Glacier Conservancy, and working with Fish, Wildlife, and Parks, Idaho Fish and Game, Northern Region of the U.S. Forest Service, Montana Natural Heritage Program, folks from Parks Canada, Wyoming Game and Fish, and Yellowstone, and dozens of volunteers. And I'm probably leaving some people out, so I apologize for that. Nearly 100 different people contributed field time to her project. So I want to just take a moment here and ask for a quick applause for all of those folks who are here and who are joining us online. I think this project is an amazing example of what it takes to do science and what we can do when we put our hands together. So Holly hails from Colorado and completed her undergraduate degree in biology at Colorado State, where she first became intrigued by research through her work with Dr. Bastille Rousseau, working on elephant movement in Kenya. Holly comes to the program here at UM most recently from Glacier National Park, where she worked as a technician and intern since 2019, assisting on projects related to all manner of critters, including fish, songbirds, wolverines, moose, bats. She tried to leave Western Montana a few times, doing stints in Nevada and Utah. That didn't stick, and we're glad she ended up back here. Holly approaches opportunities to learn and grow with a gusto that rivals my 3-year-old sponge of a human, and which is a thing that I think most of us grow out of as we get older. So I've really come to appreciate it. And she puts those new skills into action astonishingly quickly. She's done this with coursework here. Anyone knows that taking Dan's class as your first class of graduate school is the definition of jumping into the deep end of the pool. She's done this with the tips and tricks that she's learned about structuring her time to juggle analysis, writing, coursework, and fieldwork, and sanity. And she's done this with fieldwork, figuring out how to train and coordinate dozens upon dozens of volunteers and all the equipment that they need while simultaneously conducting her own field season, bushwhacking through the backcountry herself. As Holly's Glacier Park supervisor, Lisa Bate, who's here today, would put it, I could ask Holly to do anything and know that it would be taken care of to the best of her ability. So with that, it's time for Holly to share her master's work with you.
Okay, can you hear me in the back? Sweet. Thank you everyone for being here. I'm truly humbled by how many people are sitting in this room, and I'm really excited to get to share with you my master's work today. Maybe. Oh yeah. Okay, so I want to start my talk with a little story, a little bit about myself. So when I first moved to Montana right after I graduated from my undergrad. I got a job working for the USGS on their fisheries team, and I absolutely fell in love with the place. So much so that I left for a while, like Hannah said, but I decided to move back and plant myself. I figured if they can't get rid of me, they'll give me a job, right? Well, it worked. And in 2021, Lisa Bate hired me as a technician on her bird and bat crew. And on my first week on the job, we went on this backpacking trip where we had to whitewater raft part of the Middle Fork of the Flathead to cross to a stream that we were surveying to band for Harlequin ducks. And this was one of the more memorable backpacking trips for a couple of reasons. First of all, it rained the entire time, like so much rain that our rain jackets were soaking wet into the inside of our waders. Second, it was memorable for the tragedy that was my dinner falling face first on the floor of a mouse-infested cabin. However, what I'll remember most about this trip was it was my first experience working with Harlequin ducks, and I absolutely fell in love with the beautiful, charismatic sea duck. And little did I know that this trip would forever change the trajectory of the next 4 years of my life and lead me on this journey where I've gotten to work with and collaborate with over 100 different people from different agencies, countries, states, and walks of life. And this project's been way bigger than just science. It's been about conservation, collaboration, community, and friendship. And I just want to take a moment to acknowledge all of the people who have been involved in this project, because it could not have been done without so many people. And I wish I had more time to tell you about them, but hopefully you'll be able to grasp how collaborative this project's been through this project. Okay, now with that, it's my pleasure to introduce you to the critter that has the charisma to bring together so many different people. My favorite animal, the Harlequin duck. So Harlequin ducks are a small, long-lived, and slow-reproducing sea duck. On average, they live about 15 years in the wild. However, the oldest recorded Harlequin duck was 20 years and 9 months old. They don't reach sexual maturity until the age of 2. However, their reproductive success stays pretty low until around the age of 5. They're sometimes referred to as sea mice for their characteristically unduck-like squeaks that they make. And like I said, Harlequin ducks are sea ducks, so they spend about 8 months of the year out on the coast. They winter along rocky coastal shorelines, and then they exhibit a really unique east to west migration, unlike most species that migrate north to south. Also, female Harlequin ducks are extremely site loyal, and they have strong site fidelity to their natal stream. So they'll return to the stream that they were born on every year to have their young. And so this is a map from another graduate student who also just successfully defended his master's thesis at Western Washington University, but I think it just does a really good job showing the east to west migration of Harlequin ducks. That's really unique. So when Harlequin ducks are on their breeding grounds, I like to think of them like my most hardcore raft friend. They are more strictly confined to running water than any other waterfowl species in the Northern Hemisphere, which means that we find them on rivers, streams, and creeks, and not that boring still water like lakes and ponds. They're whitewater specialists, and they're immensely strong swimmers. So they feed on macroinvertebrates on the stream floors, and they can hold their breaths for up to 45 seconds and dive up to 70 feet. Also, a fun fact I like to share about Harlequin ducks is they have more broken bones than any other species, and adults often live with multiple healed fractures, which is pretty hardcore if you ask me. Okay, so now that I've convinced you that Harlequin ducks are your favorite animal, I need to give you a brief life history lesson. So there's 2 distinct populations of Harlequin ducks in the world. There's one in the Atlantic and one in the Pacific. And for the remainder of this talk, we're going to be focused on the population that breeds here in Western Montana and Northern Idaho, which, as you can see from this figure, we're the southeastmost of the Pacific population's breeding range. And when Harlequin ducks are on their breeding grounds, we can think about their breeding season as 3 distinct periods. So we have the pair season, which is roughly late April through May. So this is when the males and the females will show up and they will mate. And then we have the incubation season, which is roughly June through mid-July. And so the males actually leave the females, and when the females go to incubate the nests. And then we have the brood rearing seasons, which is mid-July through early September, which is when we would see successful broods or chicks on the stream, and they'll stay on the stream until the chicks are strong enough to fly, and then they'll migrate back to the coast to meet up with the males. And to explain why we can break up their breeding season like this, I'm going to use this screen capture from eBird to explain their breeding life history, because I think it does a good job. So they start out on their wintering grounds on the coast. Then in late April, early May, they migrate inland to breed. And we see this by this first pulse of observation showing up in the mountains. So we see the males and females. Then after they mate, the females go to incubate the nest, and the males leave and migrate back to the coast. Now, I want to highlight this behavior for a moment, because Harlequin ducks have these irregular levels of reproductive success, and a big reason is because of this behavior, because if one nest fails, there's no chance at a second clutch. And so they only get 1 chance a year to try and have a successful clutch. Then we see the second pulse of observation in August when chicks are being born, and then the chicks are strong enough, they'll migrate back to the coast. And so some of the major threats to Harlequin ducks in the region include climate change, impacts on stream flow, which can be especially harmful. So this graph is conceptualizing kind of how stream flow can be harmful to Harlequin ducks. So on the x-axis, we have the different seasons or different parts of the breeding season. And on the y-axis, we have stream flow. So on a normal year, Harlequin ducks typically begin incubating their nests right about around peak stream flow. However, when we see years where stream flow shifts later in the year, we see nests being washed out. Or on the contrary, when we see peak shift earlier in the year, we see streams drying out during the critical brood rearing period when chicks are on the stream. And apart from just climate change impacts to Harlequin ducks, anthropogenic threats like the tremendous increase in river-based recreation, summers are getting hotter, more and more people are spending time on the streams, and habitat loss from human encroachment and development are also major threats to Harlequin ducks. And so now for the remainder of this talk, we're going to be focusing on the incubation and brood rearing period. And while the males are certainly striking and charismatic, their time in Montana and Idaho is relatively short. And so when it comes to understanding reproductive success and informing conservation efforts, it's really the females that are critical to study. And so that's who we focused on for this study. Okay, so let's talk about Harlequin ducks, specifically here in Montana and Idaho now. So this map is showing all of the Harlequin duck observations we have in Montana and Idaho from from 2009, I guess from the early 20th century up to 2009. And this map is showing all of the observations of Harlequin ducks from July to September, so that brood rearing period and incubation period from 2009 to 2024. And as you can see, we've lost a lot of observations of Harlequin ducks, especially along this Montana-Idaho border. And then apart from just the loss in observations in large swaths of the region, we also have 4 long-term monitoring programs in the region. We have one in Glacier National Park on Upper McDonald Creek. We have one in Idaho on the Lochsa River, and 2 up in Alberta, one in Banff National Park on the Bow River, and one just outside Jasper on the McLeod River. And all 4 of these programs have documented similarly declining numbers of Harlequin ducks over the last 20 years. And so due to the documented declining numbers of Harlequin ducks, apart from Alaska, they've been listed as critically imperiled, or vulnerable across their entire range in North America. And so this is a screenshot from NatureServe that's just showing their status by state and province. And so because of their strong site fidelity, their regular levels of reproductive success, and slow maturity, it's thought that breeding streams have a very low likelihood of being recolonized once all the breeding pairs on a stream are lost, making them more vulnerable to population declines. However, here in the Northern Rockies, we have an amazing region-wide Harlequin duck working group. And this is a group of different agencies, and all these different agencies do yearly Harlequin duck monitoring surveys in their areas. They collaborate together to work on different projects with one another. And starting in roughly around 2000, they decided to start coming together every five-ish years to try and do a concerted region-wide monitoring effort to try and estimate population trends and changes. And so I got involved with the Harlequin Working Group in 2021. And through these meetings, it became clear that all these different groups had different concerns about the apparent declining numbers of Harlequin ducks across the region. However, and some of these concerns were that they didn't fully trust the survey method that we have. They didn't really fully understand the full breeding distribution. They're concerned about the populations. And so we decided to pool our resources together and try and fill in some of these information holes. And I would go to grad school and try to answer the question with everybody. And so together we identified 2 main objectives. The first objective was to assess the efficacy of different non-invasive survey methods for detecting Harlequin ducks, given they're present on a stream. And the second was to build a predictive occupancy model of breeding Harlequin ducks in Western Montana and Northern Idaho. Or in a nutshell, how to find them and where are they? Which leads me right into my first chapter, how to find them. Okay, so for this study, we're particularly interested in detection probability of different methods and how different habitat variables affect detection. And so moving forward, I'm going to be talking about detection probability a lot. So I just want to define it to make sure we're all on the same page. So detection probability is essentially an estimate of if a Harlequin duck is present on a stream, how likely are we to detect them? And understanding this metric is important to management because it allows us to more accurately estimate populations and population trends. So this is supposed to be a video, but I couldn't get it to work on this. But one of our big concerns is that we don't really trust our method for detecting Harlequin ducks. And a big reason why is that it can often look like this, just bushwhacking through a lot of hard to get through stuff. And these surveys are just really hard. You spend a lot of your time slipping and sliding your way up a creek or bushwhacking through miles of riparian brush and grizzly bear country. And because of how challenging these surveys are, even if you're doing your very best, oftentimes you're still watching your feet to make sure you're not going to trip or trying to make sure you don't get your eye poked out by a rogue willow. And furthermore, no one's ever figured out the detection probability of a single foot survey. And so we just don't really know, or didn't really know, how effective this method was. And so we set out not only to better understand the efficacy of these ground-based foot surveys, but also to investigate other non-invasive survey methods. And so non-invasive survey methods are ways of detecting a species without causing harm or stress to the animal. And so our 4 methods were the ground-based foot surveys, environmental DNA, motion detection camera traps, and time-lapse camera traps. And so I just introduced ground-based foot surveys, but they are essentially what they sound like: teams of observers walking upstream, looking for Harlequin ducks, and they rely on these direct in-person observations of Harlequin ducks. Now, environmental DNA is our next method. So DNA is in any cell that's left off your body. So that could be saliva, feces, feathers, dander, you name it. And these cells are present all over in our environment. An environmental sample can include some samples such as water samples, air samples, a swab of a plant, or snow samples. And so because Harlequin ducks are strictly confined to running water, we decided to test water samples to see if it was a viable method for detecting Harlequin ducks. And camera traps were our next method. And so cameras are really nice because they can allow for the detection of species that wouldn't be detected with other methods due to animal behavior or human error. However, cameras are not typically used on running water or on waterfowl due to water potentially triggering the camera, or also waterfowl tend to be really highly insulated, and so they don't actually have much of a heat signature to set off a camera. However, Harlequin ducks exhibit this really unique behavior where they tend to spend their day within a 1 mile stretch of stream, where they'll forage downstream and then they'll swim or fly back upstream that mile. And so due to this behavior, we thought that maybe by strategically placing cameras every river mile, we might be able to detect Harlequin ducks. However, again, because they're not typically used, we wanted to test both motion detection and time lapse to see if one of the methods worked better than the other. And so we surveyed 10 streams across Montana and Idaho that represented a variety of ecosystem and stream conditions used by breeding Harlequin ducks. And we surveyed each of these sites for 3 consecutive days in both 2022, or 3 consecutive days, 2 times in both 2022 and 2023. So each of these sites we visited 4 times. And each of our study sites was deliberately chosen because they've historically had breeding Harlequin ducks, and they've had confirmed breeding Harlequin ducks in the last 5 to 10 years. And we intentionally selected streams with high probable occupancy so that we could have a higher power with a smaller sample size to look at what drives Harlequin duck detections. And so the first site visit happened during the incubation season, so early July through early August. So when we would expect to see single females, or if a clutch had hatched, they were just barely hatched. And then the second visit happened during the brood rearing season, so 3 weeks later, which was late July through late August, and this was during the brood rearing season when we'd expect to see broods. And the reason we visited each stream for 2 different times was because we thought that the detection probability would vary between the 2 time periods. And so each time we visited a stream for 3 days, we would do 3 ground-based foot surveys, 1 ground-based foot survey a day. Then one day, observers would take eDNA in tandem with the ground-based foot surveys, and we had observers take eDNA at different spatial scales so that we could investigate different sampling strategies. And then on another day, observers would either place cameras or take them down in tandem with that ground-based foot survey, depending if it was the first survey or the second survey, and we had observers place cameras roughly every river mile. And so we completed 2 field seasons of this data collection in 2022 and 2023. And in total, 78 people went out and surveyed for 2,220 hours. We completed 118 ground-based foot surveys, collected 723 eDNA samples, and deployed 198 cameras, half of them time-lapse and half of them motion detection. It was truly a huge effort. And so we were able to confirm that all of our study areas were occupied by looking across detection methods. And if we detected a Harlequin duck with at least one of the method, we knew that the site was occupied. Now, it's important to note that although each site was occupied, Harlequin ducks were not always detected with each method. And so because we knew the occupancy at each site, we were able to use generalized linear models like this to estimate detection probability of each method. So here we have if a Harlequin duck was detected, yes or no as our response variable, and then we have habitat covariates that we hypothesized would affect detection. Okay, let's dive into some of our results. So I'm going to pause for 1 second because I'm going to be adding the mean detection probability of each method to this graph as we talk about them so that you can kind of compare the methods. But it's important to note that the unit of each method is different. So keep that in mind when you're comparing these methods. So for ground-based foot surveys, this is the average detection probability for one 5-kilometer ground-based foot survey. And we found that it was roughly around 0.5 for both seasons, and the 2 seasons were not statistically significantly different from one another. And the main thing that we found that affected if Harlequin ducks were detected or not was how many ducks were on the stream. Turns out, more ducks, more likely you are to see them. But actually, when it came to ground-based foot surveys, it turns out that observer bias is actually what affected detection the most, and it explained almost all of the variation within our data. However, recent studies with citizen science data show that training can really help to reduce the amount of variance between observers. And so the good news is that there are steps that we can take to try and minimize the amount of observer bias. Now, we did do training and we provided detailed protocol and supplemental materials, but there were still 78 different people across 6 different agencies with a wide range of experience that conducted surveys. And so moving forward, just having a more robust training program, I think could really help reduce this. Okay, so for eDNA, our sample unit was 1 single eDNA sample. And for eDNA, we found a 22% difference in detection between the 2 seasons. And a hypothesis for why I think we might be seeing this change between the 2 periods is I think it really depends on the spatial extent. Because although there may be more ducks on the stream during the brood rearing season because there's clutches of 6 or more, they're spread out differently along the stream. And so we don't know for sure how long eDNA persists in a stream, and persistence depends on a lot of different variables. And we would need to do an experiment to test that, which could be a whole master's on its own. However, since we had eDNA samples taken at different spatial sampling scales, we used a spatial independence analysis with our eDNA, and we found that our samples became spatially independent of one another around 750 to 1,000 meters within a stream. And so this most likely means that we're not detecting ducks miles and miles upstream, but rather ducks within about 1 kilometer. Ooh, sorry. And so, like I said, this different spatial scale of which ducks are on the stream is different between these 2 seasons. So during the incubation season, you tend to have females more evenly spread out across the stream, versus during the brood rearing season, you have these clumps of broods in the stream. I think this could explain why we're seeing this difference in the 2 seasons. And what we found that affected detection the most when it came to eDNA was stream flow. And interestingly, we found that depending on the season, stream flow affected it differently. And so I think potentially a reason we might be seeing this is typically during the incubation period, we're coming off of that peak stream runoff. And so DNA could just be being washed out of the system at faster rates during that time. Or on the contrary, during the brood rearing season, flows are typically slower, water is warmer, and UV rays from the sun and hotter temperatures could be degrading UV at faster rates during that season. Okay, so camera traps were our last method, and our unit for this was one single game camera deployed for 22 days. And game cameras were our least effective method, and we found 0.03 difference between the two types of cameras. And the one thing I want to say about game cameras, though, is although they may be our lowest detection method, and I may not necessarily recommend them as the sole method for surveying for Harlequin ducks, they may still sometimes be the correct tool for surveying for Harlequin ducks. And so as an example, sometimes there's areas that are just really hard to get into and not super realistic to try and do a ground-based foot survey or collect eDNA in. However, you could go in with a boat or thrash your way in one time to place a camera and then collect it later in the season. And so learning how to deploy game cameras, I think, was also very valuable still. Okay, so far I've talked about all of these methods in isolation. However, the power of this study is that we can start to think about how we can improve our cumulative detection by combining them. So for example, if we do a 5-kilometer ground-based foot survey and we take an eDNA sample every 1 kilometer for a total of 6 eDNA samples, we can increase our cumulative detection probability to 98%. And if we do the same thing during, and this is for just a single day visit as well. And so if we do the same thing during the brooding period, we can increase our cumulative detection probability to about 95%. Now, this is just one example of combining methods. However, we could combine our methods in a bunch of different ways, all of which improving our cumulative detection. This is just, however, the most effective way of achieving high cumulative detection in a single day visit to a stream. We can also increase our cumulative detection by doing multiple ground-based foot surveys, where each survey increases our detection a little bit at a time, which I just want to note that our long-term monitoring programs in the region have been doing this, and they've been doing a really good job at this. And something else I want to point out is that outside of our long-term monitoring programs, we've been doing single-day ground-based foot surveys for years, decades, actually over a century. We've been doing these, and we've just never known how effective these methods are, and managers have felt kind of skeptical. But now we have a better understanding that if a duck was even present, we had about a 50% chance of detecting them. That's like flipping a coin if we were going to see them. However, through this study, we've learned about the different sampling approaches for detecting Harlequin ducks, and we can be highly effective by combining them. Okay, and so what's cool about this is we actually got to go out and use our new method by taking 6 eDNA samples every kilometer in tandem with a ground-based foot survey and use it in our second chapter, which leads me right into my second chapter of Where Are They? Okay, so this is a map of all of the streams in western Montana and northern Idaho, and there's roughly 58,000 miles of stream on this river on this figure. And so it's our goal, and it's also just impractical and impossible to try and go out and survey all of these streams. And so it was our goal to create a tool for biologists and managers in the region to use in their decision-making process when they're going out to decide where they're going to use their limited resources to survey for Harlequin ducks. And so to do this, we're using an integrated species distribution model. And so an integrated species distribution model allows us to build a predictive occupancy model by combining existing observational data that comes from a variety of different sampling schemes to improve our estimator precision. And this is a figure from a paper showing predicted occupancy of rock bunting in Switzerland. And our goal was to produce a similar figure, but of streams in western Montana and northern Idaho of Harlequin ducks. So how do we get there? Well, first, we needed to identify the data that we had to work with. We need to think about what we knew about Harlequin ducks, and we need to identify what gaps that we were able to fill. So I want to bring us back to this map that we saw at the beginning of my talk to talk about some of the things that I'm noticing here. So the first time I saw this map, the thing that stuck out to me is we see these like pockets of Harlequin duck observations, and I was really curious why. And so through discussions with folks in the Harlequin duck working group, it became clear that a big reason why we see these pockets is it's been kind of dependent on the forest and the Forest Service's non-game biologists, and if they had a Harlequin ducks as a priority species for their forest over the last 15 years. And so some of these spatial gaps that we're seeing in the region could be because we just haven't looked for them. The other thing about these observations is that all of these observations are from either ground-based foot surveys, incidental observations, or citizen science. And so incidental observations are observations that were reported to agencies outside of a structured survey. So this could be from visitors, volunteers, or other staff. And citizen science observations come from eBird and iNaturalist, which are online platforms where anybody can submit observations, and then they're verified by other users, or in the case of iNaturalist, by photographs. And then ground-based surveys over the last 15 years are the same as what I explained in my first chapter, except without the eDNA and game cameras. And so just people walking upstream looking for Harlequin ducks. Okay, so now that we know what data we have to start with, we started to think about how can we fill in some of these spatial gaps to get more information that was representative of the entire region to build a robust predictive model outside of just these few hotspots. Well, one way is with environmental DNA. And as we learned from our first chapter, it works pretty well for detecting Harlequin ducks. And so a single eDNA sample, it can be reanalyzed for up to 8 different species. And once it's sampled and given to the National Genomics Lab, which is actually here on campus in Missoula, we're really lucky to have that resource so close to us. But they take these samples and they store it into a database called the eDNA Atlas. And then you can go onto this database and you can look at all of the places people have sampled for eDNA, and you can request those samples to be reanalyzed for your species. And lucky for us, fish people, looking at you, Phil, and Leah, if she's in here, really like eDNA. Also lucky for us, a lot of trout species share similar habitats with Harlequin ducks. So there's a lot of eDNA in the region that we are able to reanalyze for Harlequin ducks. And so we were able to reanalyze 230 eDNA samples for fish. Oh, there's Leah. And so this map is showing the full distribution of observations and eDNA data that we now have. Then we had one more full field season in 2024 where we went out and, following the protocol that we established in our first chapter, went and surveyed for Harlequin ducks all across the region. And it was another huge effort. In fact, we had 59 people out there surveying 48 new streams that haven't been surveyed in at least 15 years or ever, for a total of 711 hours, and we collected 261 more eDNA samples. And finally, this map is showing the full coverage of eDNA, surveys, and observations from the last 15 years that we now have to work with in our model. And as you can see, we made a huge effort to fill in the spatial gaps the best we could. And again, it could not have been done without the help of so many people. So I want to pause for just a second. So in this map, I took away the pink observations just because it was getting a little messy. So all you're seeing now is the eDNA we resampled and the surveys that we sampled in 2024. And we detected Harlequin ducks on 12 new streams where they've not been seen or found in the last 15 years, which I think is really cool and shows that I think it's just really cool that we got to develop this method and then go use it and it work and detect Harlequin ducks. However, I also want to point out that we sampled a lot of new streams between eDNA and foot surveys, and we only got detections on a few. And so I think that this further illustrates the need for Harlequin duck conservation in this region. Okay, so the last type of data we're using in our model is augmented zeros. So these are places where we felt certain were not Harlequin duck habitat. And we use these data to constrain our models when we are predicting across sites by fixing zeros to sites that we felt confident were saying was not their habitat. And so to do this, we use the National Land Cover Classification Dataset, which classifies land at 30 megapixels into different categories. And we use areas that we're counting as not Harlequin duck habitat, or any areas that were classified as agriculture or barren, which is all the orange in this map. And also we classified areas where we did not feel like we had sufficient data to predict out to as augmented zeros. Okay, so now that we know what data we have to work with, we need to take a step back and think about, what do we know about Harlequin ducks in this region and their breeding habitat? Well, we know that they need cold, clear water because they're visual feeders. They prefer mid-sized streams. They need cover for nesting and predator avoidance. They need streams with little human disturbance. And they need streams with an assortment of geomorphological features like deep pools, braiding, loafing rocks, backwaters, runs, and rapids. And we needed to figure out how we can remotely sense data that might speak to these features of streams at a regional scale. However, some of these things are hard to remotely sense at a large regional scale, like what we're doing. And so I'm going to take a moment to explain the covariates that we decided on, because I think it'll help make the results make more sense in a few slides when I share those with you. Also, because some of these things are really hard to remotely sense at a regional scale, so we ended up using proxy variables to explain some of these things. For cold, clear, and mid-sized streams, we use stream order. So stream order is a system for classifying rivers based on their position in a drainage network. So as an example, the water trickling out of a glacier at the top of the mountain is a stream order 1. And the Amazon River, the largest river in the world, is a stream order 12. And the way it works is that when two ones come together, it becomes a stream order 2. When two twos come together, it becomes a 3, and so on. And so stream order, we decided to use it as a proxy variable for cold water because typically smaller order streams are closer to their source and they tend to be colder, where larger order streams are buffered by other water in the system, so they're warmer. Also, typically larger order streams are more turbid, so they're less clear. And then stream order can be a proxy for mean annual flow. So like mid-sized streams are roughly stream order 3 through 6. For cover, we derived the percent tree cover from the rangeland analysis platform. For human disturbance, we use the Human Modification Index, which is a measure of the extent and intensity of human-induced changes on the landscape. For features like deep pools, braiding, and backwaters, we're using sinuosity as a proxy variable. So sinuosity is essentially a measure of how windy a stream is, and more sinuous streams tend to be correlated with more deep pools and deeper channels, where more straight streams or less sinuous streams tend to be correlated more with braiding in a river. And finally, for features like loafing rocks, runs, and rapids, we're using slope as a proxy variable. So steeper slopes means that water has more velocity, and water with more velocity has more power to move larger sediments like boulders and cobbles, which Harlequin ducks are often found in. And then we also hypothesized that slope might help us explain some stream features that we don't have direct measurements of, because sites with steeper slopes may be indicative of things like rapids or steep drops, which Harlequin ducks are also typically found around. And finally, we added one more covariate to try and get at water variability. And so if you remember at the beginning, I mentioned that males leave and how changes in stream flow can really affect Harlequin ducks with nests getting washed out or streams drying up. And so we were curious if more stable or less stable watersheds would affect Harlequin duck occupancy across the region. So we decided to use the coefficient of variation in the Julian day of the timing of peak stream flow. Okay, so just as a reminder of our data types, we had the structured ground-based foot surveys, eDNA, incidental observations, citizen science observations, and augmented zeros. Then using a Bayesian modeling framework, we used a site occupancy model using probability of site use based on these habitat covariates. And so for all the modelers in the room, how our model worked is we had two separate detection models for ground-based foot surveys and eDNA, which were then used in our occupancy model. And the reason we use ground-based foot surveys and eDNA in the occupancy model is because they're both detection, non-detection data. Then using the posterior distributions of our habitat covariates from the occupancy model, we use those to make Harlequin duck site use predictions across streams in the region. And we used our incidental, augmented zeros, and citizen science data to confine where we made predictions to. And it's important to note that our model is modeling site use because we're breaking the closure assumption. Okay, so let's talk about some of our results. So before I show you our results, I just want to orient you to the graph. So on the y-axis, we have our different covariates that were included in our model. And then what you'll see on the graph is the posterior distribution of each covariate, with the 95% confidence intervals is the thin black line and the 50% confidence intervals is the thick black line. And then our F statistic is a measure of how much of the posterior distribution is on one side of 0. And we're counting anything with an F statistic greater than 0.9 as having strong evidence of an effect on Harlequin duck site use, anything from 0.8 to 0.9 as having some effect on site use, and anything less than 0.8 is no effect on site use. And so from our model, we found that slope, sinuosity, and human footprint have strong evidence of an effect on Harlequin duck site use, and all of which were negative. And we found that the coefficient of variation in the Julian date of peak stream flow had some effect on Harlequin duck site use. And so what this is telling us is that as streams increase in slope, their probability of use decreases. As streams become more sinuous or windy, the probability of use decreases. But remember that slope and sinuosity are acting as proxy variables for stream geomorphology. And I think it's important to note that with these two variables, we're not able to say that Harlequin ducks prefer braided streams with cobbles over waterfalls and boulders, but I think it just points to the fact that geomorphology is important, and potentially future studies could work to tease apart the intricacies of these things. And then it also means that as there's more human development, sites decrease in site use. And then finally, stream order was also in our model, and stream order also had a strong effect or strong evidence of an effect on Harlequin duck site use. And our results on stream order matched historical research and what we knew about Harlequin ducks, which I think was reassuring because our last step was to predict out to other sites that didn't have detection on detection data. So I thought it was reassuring that what we found kind of matched what we knew, especially when we're about to put this to streams that don't have any information. So like I said, so then our last step was to use our incidental observation, citizen science data, and augmented zeros to constrain the bounds of where we would then predict out to. And to our knowledge, this is the first large-scale statistical model that can predict site use of Harlequin ducks across western Montana and northern Idaho. And so I want to take a moment to point out some of the things that I noticed when I look at this, this graph or this map. The first thing that a takeaway that I noticed is that a lot of places in this model that are predicting really well are really remote wildernesses, like Glacier National Park and the Bob Marshall, or in Idaho on the Bitterroot National Forest and the Nez Perce Clearwater National Forest. And so, for example, let's take a moment to zoom into this northwest corner up here. And so this area has the highest predictive occupancy of Harlequin ducks, and it encompasses Glacier National Park, the Flathead National Forest over on the west, and the Helena and Lewis and Clark National Forest on the east, and the Flathead National Forest and Helena, Lewis and Clark Forest make up the Bob Marshall Wilderness. And to anyone who's ever spent any time in these areas, you know it's no joke to get into some of these places on a trail, let alone into the stream. Like, no wonder these guys are so hard to find. Okay, so zooming back out one more time, because there's one more thing that I wanted to share, which is when we overlay this map with the historic observations we have of Harlequin ducks, we can see that areas where we've historically seen Harlequin ducks are still predicting really well for Harlequin ducks right now. And I think this both kind of validates what we knew about Harlequin ducks, but it also gives us a way forward when we're thinking about protecting these precious areas. So now that we have a better understanding of the tools we have to find Harlequin ducks, and we have this predictive map of Harlequin duck site use, how might we use this knowledge? Well, like all monitoring, it depends on the objective. For example, if we're interested in region-wide known species distribution, then biologists need large spatial coverage with high detection. Therefore, we would recommend using the predictive map to prioritize where to conduct surveys, starting with streams with high probability of site use, and then follow the protocol that we laid out in chapter 1 that yields a high cumulative detection probability, taking eDNA in tandem with the ground-based foot survey, and sampling as many streams as they can. Or if we're interested in trends in abundance, this would require counts of Harlequin ducks over time, and the only method we have that gives us counts currently are ground-based foot surveys. So in which case, we would suggest using the occupancy map to identify areas that are relevant to the landscape that we want to manage and accessible to visit repeatedly, both within a season and also across many years, and conducting multiple ground-based foot surveys. Or management is often tasked with reviewing project proposals and assessing the potential impact of projects on species of concern. And so this model can provide insights into areas where projects may be likely to impact Harlequin duck breeding streams and can help inform where mitigation would be necessary. So what's next? Well, I'm not just pitching out ideas of how this can be used for no reason. The power of this project is that it's fully grounded in management. And in 2026 and beyond, folks in the Harlequin Duck Working Group are planning another region-wide monitoring program and using the results from this study as the foundation for this effort. And although the area for this project was Montana and Idaho, the Harlequin Duck Working Group also includes groups of people in Wyoming and Alberta who have been amazing collaborators. Also, although my thesis map is again focused in Montana and Idaho, I hope that we are able to provide results and information that will benefit the entire region and beyond. And so, okay, and so with that, I have so many people to thank. First of all, I want to thank everyone involved in the Harlequin Duck Working Group who has supported this project literally the entire step of the entire process. I need to thank Glacier National Park and all the wildlife technicians and volunteers who spent countless hours with me thrashing through the woods to look for Harlequin ducks. I want to thank Montana Fish, Wildlife, and Parks, especially Christina Smucker, Allison Bagley, and Jesse Coltrane. I want to thank Casey McCormack at Idaho Department of Fish and Game for really carrying the Idaho side of this project. I need to give a huge shout out to our federal partners over at the Forest Service who did a majority of the field work. I want to especially thank Kara Staub and Tammy Fletcher for making this project a priority in this region. And I want to thank Tommy Franklin and Jennifer Hernandez at the National Genomics Lab of Fish and Wildlife Conservation for all the support with the eDNA. I need to thank Barb Lancaster. Without your support, I would not be here today. And the Glacier National Park Conservancy and all of the various people and agencies who contributed funding to this project. Bryce Maxwell, Dan Bakken, and Scott Bloom at the Montana Natural Heritage Program. All of my peers in the Boone and Crockett Lab. I feel so fortunate to have been part of such a great lab group. The Bruner Lab for adopting me as an honorary member throughout my time here. I need to thank my technicians over the last 3 years, Megan, Kai, and Annika. You were all the brightest part of my field season and helped me get through the summers more than you know. And I need to thank my committee, Dr. Kriya Bruner, for your valuable input on my work. The way you care about those around you is inspiring, and I'm extremely grateful to be a part of your lab group. Dr. Thomas Reiki, for all your guidance and statistical support. You're an incredible teacher and scientist, and you've taught me more about Bayesian statistics than I could ever imagine or dream to know. I'm humbled to be mentored by you. To my two advisors, Dr. Josh Millspaugh, for providing me with the tools and feedback that I needed to be successful. The way that you care for the success of each of your students is obvious, and I'm extremely grateful to be part of a group of people that fostered so much community and support, and I look forward to continuing to work with you in the future. To Dr. Hannah Spate, for providing me with the technical support through the entire process and challenging me to give it my best shot, even if it's messy. And for also just being a role model and showing me you can be a great scientist, mom, and athlete. And I need to thank my supervisor, mentor, and committee member and friend, Lisa Bate. You've been the most incredible role model, and it's because of you that I'm here today. You've shown me what true passion for the natural world is, and I'm honored to have been someone that you've mentored and brought into it, and I hope I've made you proud. And finally, I just want to thank my friends and family, my mom, my dad, both my brothers, my partner Mason, and all of my friends who've supported me along this way. I could not have done it without all of your support. So with that, thank you.
Got time for 2 questions.
Kelly.
I'm curious with stream order as a metric for habitat or habitat in habitat. I'm curious if you were able from your model to get an understanding of the explanatory power of that variable, and then also if you consider using something that maybe a little more consistent as a variable, such as like upstream drainage area, just because when you have 2 first order streams coming together, a second order stream in one place can be very different in another location.
Yeah, totally. So we found in our model, so the question online for those online was if we like how explanatory stream order was in our model, and if we considered using any other variables for like stream size. Yeah. So stream order we found had a really strong effect. So I think the F statistic for it was greater than that 0.9 threshold that we were counting anything with a strong effect for all the stream orders. And we only use stream order 1 through 6 in our model because we didn't have any data on stream orders larger than 6. And so we didn't feel like we had enough information to accurately predict to streams larger than that. And as far as using other metrics, we did not, mostly because streams are complicated and I didn't know to think about that, but that could be something to think about for moving on. So thank you.
So awesome talk. I wonder, so you might know there's more ducks out there. You're telling us there's lots of ducks we can't detect very well because they're isolated. They're also really well protected. They're wilderness areas. There's not a lot of human disturbance. The thing that can change up there, you talked about in talk, is these attenuations and stream flows along that species. What do you think the next steps are in context of how those effects could be impacted, whether it's the changing stream flows, hen survival, brood survival, which actually might be affecting the population, would build on your work? What's that next step?
Yeah, so for those online, the question was, a lot of the places that are predicting really well are already protected from wilderness and are remote. And so Patrick was asking like maybe what the next step is. So I think a next step would maybe going to see if some of those places where we haven't seen observations in a long time but are protected, going back and resurveying them and seeing if they're still occupied to try and start to look at like, are those streams blinking out? Because a big question that the Harlequin Duck Working Group had is kind of changes over time and like, are we losing these populations and are streams blinking out? And we just don't know. So I think maybe one of the first steps could be to go and investigate. places that we knew where Harlequin ducks were.
Everything about looking at your stream flow trends over time. So like, are you moving towards a new normal context of where you're
Oh yeah. Yeah, totally. Cool. Thanks. Yeah. And then we'll come back to you, Chad.
So looking at your models, it looked like all the effects kind of followed your hypothesis in general, except for it looked like slope. It looked like slope kind of had the opposite effect of what you anticipated. Could you speak to why you think that might be?
Yeah, so slope was derived over the whole like 1 kilometer or 1600. So our sites were 1600 meters, so roughly a river mile. And so slope was derived from the whole river mile, so top and bottom. And I think, so I'm kind of just speaking from experience of like doing some of these surveys, but there's a couple surveys that we did where it was really steep, where it was just like a raging cascade the whole way. And I remember being on this survey being like, there's no way a duck would be here, because like, although they prefer these steep slopes, or like they prefer like drops and cascades and things, they also still need those deep pools and backwaters and refuge. And then also when we look at hold everything constant in the mean, our mean slope was actually like really high. And so I think they tend to prefer, like they still need the streams with those refuge areas, which like makes the slopes over the whole 1 mile stretch like maybe less steep. Yeah. Chad.
My question might be related in a way. I'm just curious, you talk about strong site fidelity to their natal breeding ground. And so if you do have climate change tied into these things, do we know how adaptable or plastic they are in the behavior when they return? Like if they go back to their natal stream, are they, do you ever see them go miles away from where they were? Or are they really locked into a
I don't know for sure. Lisa Bate might be able to speak more to that if they migrate farther up. I think what I've read, they tend to stay, like come to the, once they establish like their breeding area, they tend to come back to that same area. However, when they're still young and like they'll come back to their stream, but they might go up and downstream when they're establishing like where they're breeding. I don't know for sure though.
We did see some females move up or downstream if they failed. But if they were successful, they often would even pick the same nest. But there's a lot of unknown. But I mean, some of those females would move 10 miles, but then when they'd settle for incubation, it sort of mean that's it.
Yeah. Yeah. Jonathan?
I was really curious, like the eDNA stuff, so you looked at like a single detection, right? Was there like, could you increase the number of number of samples, and does that increase your eDNA detection? Like, was there a certain point where it matched or exceeded to observation surveys?
Yeah, totally. So you can get the same really high detection probability just using eDNA samples. However, because you still need to take eDNA samples like throughout the stream, you still essentially have to do a ground-based foot survey. And the power of doing a ground-based foot survey in tandem with a foot survey is you can get relative abundance data because you're actually counting them, where with eDNA, it's just detection, detection, non-detection data. And so that's why we typically, we would recommend also doing the ground-based foot survey, because you can get also this extra quality type of data. Yeah. Christopher, and then we'll coach.
I'm curious, just having spent 2 years, I guess now, studying these ducks, maybe more than that, I guess. Like, what do you think some of the implications of your research are for storytellers in the humanities and public media more broadly as a result of everything you learned?
Oh gosh, put me on the spot. Yeah, so I think one of the most amazing parts, also, sorry, for the folks online, I haven't been repeating questions. Christopher asked what he thinks if my research has any implications for more like humanities and storytellers and Harlequin ducks. So I think the thing that comes to mind with that is through this project, I've gotten to collaborate and work with so many different people. And I've had a positive experience almost the whole way through, which I don't think is very common. I think there's always people on both sides, but people really love Harlequin ducks. And I think part of it is because, well, one, they're not a game species, at least here in Montana. But then also they're really beautiful and charismatic. And so I think people really latch on to that. And so they do have more like cultural value in this region. And across their whole region, they have cultural value. And as well as like economic value, people flock to places to come see them. Like most observations we have are on Upper McDonald Creek. That's a big draw to people coming during the hiker biker season. And so, yeah, I think like just their like charisma brings a lot of people to like really loving them. So,
I'd like to take one more question.
Okay.
Yeah, I kind of follow up on the site fidelity question because
Yeah.
I was struck by how there was a lot more high quality habitat than like known occupied sites, but also you said they have extreme site fidelity, like they're up and down the river, but they're not going to.
Yeah.
So say we were able to successfully expand the population, how do you think they would be able to find these new streams, or is it like you say translocations would be needed, or how like if one winks out and then that stream is gone forever because no harlequins remember where it is, like how would.
Yeah, that's a great question. For those online, he's asking about site fidelity and if a stream were to blink out, if we could like translocate them or if how they might expand into new streams. That is a great question. And I think that's something we've talked about a lot in the Harlequin Duck Working Group because they do have this strong site fidelity. And as far as I know, we've never done any translocations of Harlequin ducks in this region. So we don't know how well they would work and if they would work. Yeah, I don't know if that's, yeah.
Thanks,
awesome.
everyone.
Thank you.
I would be happy to answer many more questions about Harlequin ducks at Kiwanis Park at 6 p.m. right after the sunshine.
Yeah. Yeah, and thank you everyone online for coming to my presentation. Oh yeah, is there?